Molecular techniques have revolutionized horse management and health and have provided practical solutions to complex problems in the various horse disciplines internationally. Professor Alan Guthrie, Director of the Equine Research Centre in the Faculty of Veterinary Science at the University of Pretoria, recently hosted a lecture on this exciting topic. The Plaas TV team visited him afterwards at Onderstepoort to find out more.
I was privileged to present expert lecture at the University of Pretoria, the 34th expert lecture. Title of my presentation was Harnessing Molecular Techniques for Horse Health. What do we mean by molecular techniques? It's basically or mainly looking at techniques that look at nucleic acid. So DNA in animals, and then when you get to viruses and animals, other things like RNA, which is used to translate what the genetics codes that we've got in our DNA and make proteins. So it's basically the molecules inside cells and analyzing those molecules to understand things that are happening either in cells, in animals, or obviously in infectious agents, so viruses, bacteria, etc., etc. So the new techniques can be applied across all species and help you understand some of the things that are happening either in totally healthy horses, and for example, why the horse is grey. So that you can do a molecular test to show, yes, that horse is definitely grey. It's got the gene that is responsible for horses turning grey. And so there's certain horse breeds, for example, the Appaloosas, that don't like grey in them. So they want to work out very early on that they aren't carrying the grey gene, and therefore that later on they're not going to breed grey horses because the grey gene is dominant and it may be expressed. So those are a few of the things that's what we're looking at, the principles of what we're looking at. If we look at the different techniques, we look at animals, for example. The genetic fingerprint, the DNA fingerprint of animals is very common and the techniques have been in place. Well, historically we used to use in horses blood typing. That was replaced in the 1990s with DNA typing. From the DNA typing, you can verify that the pedigree of an animal. If you look obviously at sports horses, so race horses and various other sports horses, the value of the animal is dependent on its pedigree because of how its parents actually performed. So if you take the thoroughbred horse, for example, the thoroughbred industry worldwide is supervised, or one of the supervision sides of it is the International Stud Book Committee, and that sets the guidelines for how you register a thoroughbred horse. And every single thoroughbred horse born after the 1st of January 2002, anywhere in the world, has to be parentage verified using DNA typing prior to being registered. So if you cannot verify the parentage, they're not registered, they're not a thoroughbred horse. So that's a requirement of that breed registry worldwide. So that is important. But if you look at the newer technology where things are moving now, so if you look at, and it's moved the quickest in production animals, where you're looking at specific production traits. So you're not only looking at parentage, but you're also picking up other traits. For example, muscling, and one of the more interesting ones that I understand people are now paying more money for, is they've even got a marker which tells you whether your cow is going to produce less greenhouse gas, and therefore is more of an environmentally friendly cow to have in your herd. So those are things that you can do with the more modern techniques. Yes, people are spending a lot of time looking for markers for various performance traits. Is it a horse that runs very a sprinter, so very fast over a short distance, a stayer, a horse that's going to be better over a longer distance, so markers of that, markers of being able to have the explosive power necessary for jumping, other animals that may be better at things like dressage. So yes, there are markers of that. Obviously markers for horses that are can do the different interesting gaits. So that is something that you may want to select for, because the animals can give you the gaits that you're looking for. Obviously colors are a thing that some people look for. So in their breeding program they may include markers to look at specific colors and breed towards those colors. Obviously in horses as well, we've got various genetic diseases. So they're usually breed specific, and these markers pick up your different genetic diseases. And the one thing that people look at is they say, okay, my horse is a carrier for this particular genetic disease. That is not a life sentence. So they're looking at the genetic background to more complex situations like kissing spine. But if we take lavender foal disease in Arabian horses, okay, that is a recessive, like a lot of the diseases are. And so you need both the mother and the father to be carriers for a foal to be an affected foal. So yes, if you take any horse that is selected to be a breeding stallion, they have a whole lot of wonderful, unique traits that make them be selected as a stallion. Yes, they may be a carrier for one of these things, but if you are careful in your breeding program and you only mate mares that are not carriers, you essentially you may be breeding some carriers, but you're not breeding affected animals. So it makes a difference in your herd over time. And if you're wanting to, if you think that particular breeding was good, you can always breed another horse and get potentially another stallion that is not a carrier. Okay, so there's no reason, it's definitely not a reason to get rid of animals from a herd. You basically need to use it to guide your breeding program. Yes, temperament is something that one obviously can look at, and have horses with a better temperament for certain activities. Yes, so that's all in the new systems that are being produced now, and it's called genotyping by sequencing, is coming online and should be available, and with time you'll just get more and more information about your particular animal, just like they're getting for the production animals now. All right, African horse sickness. Obviously, it's caused by African horse sickness virus. So we've been applying genetic or molecular techniques to African horse sickness for our program started here in the early 2000s. And the first thing that we wanted to do is develop a rapid test for testing for African horse sickness virus to see if an animal is infected or not with African horse sickness virus. So I think few years ago, if we'd been talking, not many people would know what a PCR is, whereas since COVID, most of us have had a PCR done on a sample collected from us because of the COVID. So there I think we all realize that you can have a sample collected, within a few hours you've got a result. That's the huge advantage of your PCR testing. So we looked at viruses that we had stored in our freezers here and analyzed them to see what sort of diversity there was in those viruses. We started, there are nine types of African horse sickness virus. So we looked at all 9 types, and then we looked at another gene which varies from virus to virus, or African horse sickness virus to virus. So we got a diverse set of viruses, and then we started looking for places inside there that are the same in all the different viruses. So we identified the 3 most likely genes, and then we went and looked at those genes and then identified the test which would work the best, so that we could pick up all the different variants using one test. So that test we developed in the early, just after 2010. But then the results we were getting were good enough to say, well, hell, this is a test that should possibly be applied for international trade, for testing for horses if they're going to be moving from one place to another. Yeah, the World Organization for Animal Health basically provides a manual of tests which they accept for international trade purposes. They give guideline on how you should show that a test meets their requirements. We put all the data together that's required for their validation, and in 2016 they included it in the manual for tests which should be used internationally. So if you look at laboratories worldwide, there are probably 50 odd laboratories that do testing for African horse sickness, and all of them use one of the two tests or both of the tests that are listed in the World Organization of Animal Health manual for testing for African horse sickness. What difference has it made? If we go back and look at African horse sickness outbreaks in South Africa in the 1990s, if you had an outbreak of African horse sickness, you'd collect a sample, you take it to a laboratory for virus isolation. It would take somewhere between 7 and 10 days to be shown to be positive, if you're lucky. The next thing is, if you want to start vaccinating, which bottle of the vaccine do you use? Because you've got different types in the different bottles. So you type the virus and you say, okay, we're going to go in and use bottle 1 or bottle 2 of the current vaccine. So that additional test would take another 7 to 10 days. So you're looking, if you're very lucky, 14 days. If you're not so lucky, 3 weeks till you've got an answer of which virus you're dealing with. Your program, if you say, well, hell, we can't wait 3 weeks, we're going to start vaccinating today, it's a spin of a coin. Have you chosen the right bottle or not? Half the time you'll be right, half the time you'll be wrong. Whereas now, you get a sample within a few hours, somewhere between 3 and 4 hours, you've got a result. And not only can you work out that is definitely African horse sickness, there's additional tests you can go in and do which actually type that virus. So on the day you get the sample, you can turn around and say, yes, it is African horse sickness and it's type 1 African horse sickness. Therefore, if you're going to vaccinate, you use other horses in the area, you use bottle 1 of the vaccine. So it has made a difference to the time it takes to do the diagnosis and understanding what types are circulating in different areas, so people know if they've got, for example, one of the things you always got to consider is if we take when African horse sickness occurs in South Africa, it starts in this part of the world, February, March gets a bit more, April a lot worse, May, beginning of May is probably the worst time, and then it suddenly drops off after that. But if you get an outbreak of horse sickness in an area where there are a lot of young foals, so places where they're breeding horses, at that time of the year, you've got lots of foals which have never been vaccinated and obviously would have got antibodies from their mother, but have now lost those antibodies, so they are totally susceptible to the virus. So you've got to weigh up your options. Does it make sense? And here you'd obviously need to consult your veterinarian, and you weigh up the risk-benefit ratio between vaccinating in the face of that outbreak and not vaccinating. But it is an individual decision that needs to be made by the breeder and their veterinarian on what the risk-benefit ratio is. Obviously, they could apply general control measures with stabling and putting up netting and insect repellents, etc., and insecticides. But that's an individual decision depending on the value of the animals. Yeah. So then, also the other thing going on from there, if you look, and I think most of us experienced it during COVID, in that within a fairly limited time, people spent billions of dollars making vaccines, and a lot of us were vaccinated against COVID, and I believe it made a difference in where we are today. If we hadn't had the vaccines, I think things would have been very different to what we have today. So yes, new technologies are also being used for vaccine development. There are some exciting things for African horse sickness virus. If you take our current vaccine, the first version of that was produced in the 1930s. In the 1960s, they started to update it. So we have version 2 is 60 years old, and African horse sickness vaccine has been around for 90 years. It has made a fundamental difference to African horse sickness in this country. I don't believe we would have the horse industries in this country if it wasn't for the current vaccine. I'm not saying there are no issues with the vaccine. I'm just saying it has made huge differences to our industry. I think one of the consequences of COVID is that the description of the molecular techniques are widely available for one, diagnostic testing, second is vaccines, and third is how the body reacts to the virus when they're infected. So actually understanding of disease. There are a lot of good lay articles, and so I'd say probably a start would be, if you're interested, consult Professor Google and go from there and get some information.